thank you all for coming out again. This is our first meeting after our big event in February, which was the mushroom fair that we had at the LA Arboretum in Arcadia. So if you attended that, I'm sure you enjoyed some part of it, at least. But no, it was a great event. I enjoyed it a lot. I think it was quite successful. And as you can tell, it's quite hot outside, so we don't have many mushrooms. We don't have any mushrooms on the table right now. But we have some interesting intellectual offerings today on this subject. And so this is our March meeting of the LA Mycological Society. We meet here on the third Monday of the month from October through May, except for February when we have our annual fair. And in addition to having a guest speaker each week, we usually start off with mushroom identification at 6:30. So if you have any mushrooms next time that you'd like to have looked at, we could do that. Just some notes about the room. We do not use microphones because the acoustics here tend to be good enough that voices can carry. But because of that, if you're talking to someone beside you, everyone can hear it. So just be mindful, even with a very strong whisper, the rest of us can hear it. And with that said, is there anything anyone on the council would like to address? Okay. And now I'm very pleased to be able to introduce our guest speaker today, newly minted PhD from UC Berkeley, now making her home here in Los Angeles, Grace Stark.
Hi everyone, thank you so much for inviting me. As Rudy said, I live here now, so it's great to come back and give a presentation. This is actually very similar to a title of presentation I gave for LAMS in 2022, and that was when I was really first starting my PhD, so I'm excited to tell you how I kind of wrapped it up, and yeah, let's get started. So this is sort of the two photos I chose for my title slide because I'll be talking to you later on in the talk about, just can't see me, okay. Neurospora crassa, and a lot of it is cell biology of how fungi perceive bacteria and mount a response. But I also have, one of my chapters of my thesis was a review on all microbial pressures on fungi and basically trying to solve the problem of why we haven't discovered fungal defense systems and the cellular mechanisms behind fungal defense systems. So a little bit about me. I started off in a fungal ecology lab in college. I worked in the Hawks lab, she's now at North Carolina State. And she is very interested in the endophytes of grass. So this is a Texas native grass, Aristida purpurea, and all of the fungi, some of the fungi I extracted from it. Notably, I also extracted xylaria, and it would fruit on the plate, and that was always fun. Yeah, so I learned a lot about extracting metabolites and doing HPLC-MS, and also how to do ecological studies, ITS, and sequencing. I then decided that I wanted to study more molecular biology because I was a biochemistry major and I was getting really interested in cell biology. And so I started in another lab in my undergraduate, the Barrick lab, and I studied a very common agricultural plant, Basiolus vulgaris, and its interactions with viruses. And so this is me with the plant and inoculating viruses onto the leaf. And that's when I was first introduced to the plant immune system, which is kind of bizarre to me because, you know, you only hear about the human immune system. You know, we all know about our own immune systems, but this was at the beginning of, actually, I mean, yeah, there's this idea now that everything has an intricate defense system, and yeah, there's actually a lot known about plants because of them being in our agriculture and needing to be protected against blights and pathogens. So there's a lot known, and that was a really cool introduction to everything having an immune system. I then went back to fungal ecology, my two interests pulling me. This was a fire fungal ecology lab, the Bruns lab up at UC Berkeley. It was my first rotation, and it was awesome because I immediately got to set a forest on fire when I got to California. And it led to this publication where I did RNA sequencing and some carbon isotope tracing, looking at how the post-fire fungus Pyronema digests charcoal. So that's the name of the paper if you want to look into it, but I'm not talking about that. I then worked back on the plant immune system. I switched back into a lab, another rotation project for graduate school, where she had mostly studied wheat and Arabidopsis immune systems, but one of the graduate students in her lab started studying duckweed and how duckweed had different immune receptors, and we started looking into how it differs from other plant systems, and it's also very easy to grow in the lab in a petri dish and infect with different bacteria. So it was a good pathosystem, so we published on that, and there's another paper that's part of my thesis, but I'm not talking about duckweed. The main thing I'm talking about today is if fungi have an immune system. So I've been studying now the plant immune system and fungal ecology. I started thinking, well, do fungi have a defense system? Do we know about this? And that led into my thesis work. Yeah, so I have a homing slide because there's a lot of information. The first section I'm going to talk about just quick info on immune systems across the tree of life and what we know to orient people, but a lot of these ideas and ways I'm talking about this is actually a review I wrote that eventually I will publish, but is actually published online right now as part of my thesis, which was published last week. I got the notification, and now when I look up certain things related to my thesis pops up on Google, which is cool, but I'm also like, oh no, I don't need that. And then the second section is going to be the accumulating evidence for the fungal immune system, which is more dispersed, and yeah. And then the third section is going to be the work I did on Neurospora crassa and bacterial interactions, which is sort of the system I designed and I had access to in grad school to basically answer a lot of these hypotheses on the fungal immune system that accumulated. Okay, so a little primer. Cells are constantly interacting with their environment and each other. This is a very, very basic diagram of cell biology, and where there are receptors, there are these proteins made by genes, and they locate to the cell membrane and the cell wall, and they sit on the outside of the cell, and they recognize extracellular ligands like food sources. Is the most commonly studied for fungi, like simple sugars, like how fungi respond to different sugars and complex carbohydrates as they're degrading. That has been really well studied, and the receptors that dictate these metabolisms the fungi then turns on or off. Light is a very common one, like photons and receptors. And this is then usually translated through MAP kinases, transcription factors, these signaling molecules that then turn on or off certain cellular processes, typically in many different ways. This is not really showing, but it can be the transcription of a gene, it can be the destruction of a protein. And these are oftentimes these cellular feedback loops that happen. And so this is very basic, but The other thing that cells do, especially when you're a multicellular organism, is you have cellular crosstalk. And so this can be internally if the cells are connected, like they are with hypha. It can be externally, as it is with us and a lot of other cells that are in consortium together. But what I'm going to talk about is the ligands of other organisms. And the receptors that typically dictate those responses are actually our immune receptors. And so a basic question, and a really actually difficult question to answer, is what are immune systems? And you know, people can say, oh, they're protective systems, right? But when you look all across life, what it tends to boil down to is these non-self-recognition systems, and they are there to maintain homeostasis from invading organisms. And so this refers to the fact that you have your own autonomous organism, and if another organism comes and tries to utilize your cells, your body needs to be able to recognize first that that organism is not yourself. And a lot of organisms, like microbes that try to invade your cells, like viruses and bacteria, they try to quell these responses that are instigated. But it's typically, I'm getting slightly ahead of myself. Let me just explain how our immune systems typically discover. The main one is looking for a cure for a disease. And this is how the immune system in mammals, in us, where we get all of our basic immune concepts come from. And so these are antibodies that are made by specialized cells in us, B and T cells, that can evolve somatically to recognize different epitopes of a virus, right? And so this is what vaccines take advantage of. They take advantage of our ability to make these recognition molecules that can recognize when a certain virus is around and what kind of response and defense they need to mount. And this is similar for many different layers of the immune system. It's usually about recognizing that there is an invader, and then that can start a defense reaction. The other way that immune systems are typically discovered is homology of known immune components. So we knew about the adaptive immune system, these B and T cell biology for a long time, ever since the discovery of the smallpox vaccine. But then researchers that were studying Drosophila melanogaster, which is a model organism for development, like larval development, they found that one of their developmental genes, this toll gene, was actually required for antifungal defenses. So they observed that when this gene was knocked out, Drosophila was much more susceptible to fungus, specific fungus that wants to kill it. So this then actually informed human biology, because we then realized through looking through the genome that we had these receptors as well. We had very homologous receptors. And then that led to the discovery of some of the core innate immune genes that are found in white blood cells and macrophages. So this is where we kind of start mostly talking about the innate immune system that is shared across the tree of life. Yeah.
Just to make sure everyone's on the same page, can you define homology?
Right, yeah. So homology, there are many different forms of homology, but homology, what is the actual definition? Basically, a basic definition is if you have a protein or a gene, it is very similar sequence-wise to another gene. And typically, they have evolved from that gene. And it's very similar to building a tree of organisms. You can build a tree of genes and say, oh, we have these genes that are very, very similar in sequence to the genes of Drosophila, right? And so if we can study, and especially mice, right? A lot of our genes for us have been discovered in mice because we have very high sequence homology throughout the whole genome, and so each gene is very similar. You know, they're different in ways, and typically, like, when we're studying genes in model organisms, you actually take that gene from human, you put it in mice to actually try to study that gene. But they're very related. Like 2 organisms are related. These 2 genes are related. And some genes are more related even at distant, like an ape and a fruit fly, right? They're quite distantly related, but the genes can be anciently conserved. Like this is true for our RNA, which builds our ribosomes. Like that is something that is conserved throughout all of life. We all have ribosomes, and it doesn't really change too much. Is that helpful? Okay. So basically, what was found in fruit fly, we now have really wonderful mechanistic understanding of all the TLRs in humans, all these toll-like receptors. So there is 1 through 9 on the screen, and they're known for being pattern recognition receptors that specifically recognize microbial-associated ligands on the surface of the cell and on the endosome. So they're typically membrane spanning. And these proteins recognize very conserved ligands that can be found in all microbes that aren't mammals. So flagellin, most of our cells don't have flagellin. So if there's flagellin in the presence of us, that usually signals that there's a bacteria, right? And we need to turn on our defenses for bacteria. So that's one of the most common ligands. Lipopolysaccharides and triacyllipopolypeptides are all like very specific conserved bacterial ligands. And so these are very general responses that are turned on when these ligands are, recognized by these receptors. And so this is very similar to if these receptors are recognizing that there is sucrose in the presence of the cell. So the cell wants to upregulate its sucrose intake. It wants to upregulate its ability to metabolize sucrose. It's very similar, like these very common bacterial ligands alert the cell that the cell needs to probably turn on a defense mechanism against bacteria. And so a lot of the ligands in the endosome is after autophagy, where the macrophage, so these proteins in us are most commonly on macrophages and white blood cells that survey all of our tissues looking for bacteria, right? And then they'll engulf the bacteria and then they'll destroy it typically in another vesicle in the cell, and this is called the endosome. Once the bacteria is destroyed, it typically leaks a bunch of DNA, RNA, and so this can instigate the reaction further. There are also internal receptors, and this is actually the receptor that I'll be talking the most about because these have been discovered in fungi. And so these receptors recognize invasions of microbes inside the cell. So there's a lot of microbes that go inside of your cell to try to manipulate it. I think Listeria is one of them. Listeria goes internally. And so it bypasses a lot of the external receptors and it mitigates the response. And there are other receptors inside of the cell that try to recognize and turn on responses. And so this is showing after the recognizing of this intracellular bacteria, there's this specific mechanism that's turned on. And after this signaling mechanism, a repressor of a transcription factor is degraded. And so this transcription factors go into the nucleus typically. They'll wait around repressed. Similar to the receptor, they'll wait around repressed until they have their signal to be unrepressed. And then they'll rapidly do a reaction. And so this is showing more of the pathway, and yeah, you won't be tested on this. But yeah, the ligand binding triggers defense through immune activation. So this is sort of how it triggers defense. These transcription factors are really well studied human transcription factors, in our immune system, and innate immune reactions, NF-kappa B, that turn on these cytokines that can turn on even more genes, that it's just like cascading effect to create a pretty intense reaction, because the faster you kill the microbe, when it's really, just one microbe before it becomes, hundreds of microbes, the better. So these reactions are as fast as when you feel, heat or a pinch. Like they're very rapid cellular reactions that can alert a cell it's time, to change and turn on antimicrobials, turn on things that will help defend against, the bacteria in my presence or the virus in my presence. So the intracellular receptors I introduced, which I didn't give a name, which you probably should, they're referred to as not-like receptors. The name is debated, and from here on out I'll refer to them as NLRs. They are in the news as also inflammasomes because they make this multimer protein that typically injects itself into the cell wall and causes calcium influx and programmed cell death and apoptosis. And when these proteins are not working well in a human, there's typically an autoimmune disease. So lupus is caused by an NLR being mutated. And another autoimmune disease I forgot about is also caused by an NLR being mutated. So these are very much triggers of immune systems that can cascade into, very large cellular effects. Another thing, and what I just kind of referred to of them being very important is to induce protective programmed cell death, which is especially resourceful when the pathogen you're defending against is a obligate biotrophic pathogen. And so I'll tell you what I mean by that with an example. So powdery mildew, a fungus, and the ascomycetes is an obligate biotrophic pathogen. It requires the plant to live. So it's not going to live freely in the soil. It requires to infect a plant. And so this is showing when an NLR is present, you see a very healthy plant crop. And then if you looked at it microscopically, wherever a powdery mildew chlamydia landed on the cell and tried to infect, you would see cell death. And so this is a specific dye that you can dye cells with that shows death. And the same color is showing chitin in the next one, so it's not as distinctive, but they're two different dyes. Without the NLR, the plants are dead, and if you look microscopically, you would see that the powdery mildew was infecting the leaf. And so this is the hypha of the powdery mildew and a tostoria as it's infecting the plant leaf cell. And so this is also alluding to the fact that plants also harbor NLRs. But what I just talked about with animals, this is the animal example. So this is your liver, and with the NLR, you have a healthy looking liver, no spots of bacterial infection, which are what these are. If you look at it microscopically, you would see sheathing of the liver cells, you would see apoptosis of the liver cells, because wherever the bacteria attempted to infect, the cell had the reaction to kill the cell, so it would protect the other cells around it. Because you can replace one or two cells if you're a multicellular organism. So that's honestly one of the most common reactions that NLRs do. But if you looked at the healthy liver, you would see the cells intact. But if you zoomed in, you would see bacteria infecting them. So the cells are living, and it's being manipulated by this bacteria. And this is also very common with viruses, because viruses are obligate on us and everything, right? And any virus that infects requires a host to live. Okay, so like I just alluded to, the plant innate immune system is very similar. And so again, you have these pattern recognition receptors on the cell surface, and then you have these pattern recognition receptors internally. What this is showing that I didn't show with the animals is that typically the pathogen has all of these offensive strategies to quell this response. This response And so a lot of like pathogens and hosts are in this sort of arms race. Like they'll evolve a way to knock out an NLR or the genes being transcribed in some way, and then the host will figure out a way to recognize that before it occurs. And so this evolution, this way that organisms have evolved in competition is like prevalent throughout all the tree of life. And so this is my figure from my thesis. It's kind of small, but this is just showing that TLRs, these cell surface receptors, and NLRs are pretty conserved across all of life, especially NLRs. And they're in this class of signaling stand ATPase proteins that have ancient origins in bacteria. And recently there has been discovery of NLRs, like very homologous NLRs in bacteria. And so it's thought that these innate immune systems evolve basally in the tree of life, which, you know, corresponds to the fact that as an organism evolves, it needs to be able to recognize that it's its own autonomous organism from its environment and other organisms. So this non-self recognition is sort of this just ancient fact of life. Like once you become an anonymous organism, those cells decide to work together. You distinguish who is not trying to work with you. So why study immune systems? And this is sort of where I came from in Berkeley. Like immune systems are very hot in Berkeley because Jennifer Doudna is at Berkeley. And her discovery of CRISPR was how viruses defend themselves against bacteriophage. Bacteria defend themselves against phage viruses. So bacteria themselves have viruses infecting them, and CRISPR is actually one of the systems in bacteria that, you know, renders that bacteria immune to another virus attack. And so, yeah, this study is really, it's not as recent anymore, but it goes into how we categorize defense systems and how it's getting really hairy as we're like figuring out more and more defense systems, especially in bacteria, because bacteria are prokaryotes, they evolve so rapidly, and so do the viruses. So they're great systems for discovering like how an organism evolves defense systems and how the competing or offensive phage evolves offensive systems. So yeah. Sorg, he's great. Okay, so now I'm going to go on to the accumulating evidence for the fungal immune system. So do we have any questions on immune systems? It mostly just boils down to like being able to recognize this microbe with this microbial ligand and mounting a defense. Yeah.
Is it the same for passive and active immune systems?
Yeah. What do you mean by that?
Like, say, a goldfish has a passive immune system, where humans have an active and a passive. Where
Right.
a goldfish will respond when they're exposed to something, when they're not exposed to something. They won't necessarily have, their immune system will go away. They're a lot isolated from a lot of pathogens of other fish.
Oh, I see. So they don't have receptors, or?
That's kind of my understanding.
Oh. Yeah, I mean, my understanding is that at least zebrafish have immune receptors, like this innate immune. But it evolves that recognition, like the innate immune recognition evolves through generations. And so the adaptive immune system is in mammals and higher vertebrates, and that evolves in real time, like somatically. So yeah, if the goldfish is not able to mount a defense, it probably won't be able to in its lifetime, and it will die. But yeah, and then that selects for the goldfish that are able to defend, and then
Or
those.
are exposed to it more often,
Yeah,
so they can maintain it.
yeah. Yeah, I mean, I know of like passive, people think of passive immune systems as like just constantly transcribing antibiotics and stuff, like, but I don't know of the term passive and active immune system, but yeah, I'll look into it. Yeah, so when I first started my PhD thesis, as I just explained in the beginning, I wanted to know what immune defense systems fungi have. Interestingly enough, okay, first when you look this up on Google, you will get how do humans defend against fungi? What is the immune system like for humans defending against fungal pathogens? Of course, because that's the main study we have is human biology, right? But then, luckily, a little deeper you dive, you'll see that actually these systems building the mycelial network is dictated by a lot of these immune receptors found in us. And this is great because one of the emeritus professors working at Berkeley studied one of these organisms that was foundational for discovery of how the mycelial network forms. And so basically what happens, when a spore falls, the fungi radially grows outward, right, to cover as many nutrients as it can. And then as it's radially growing outward away from each other, as the internal hypha then make cross connections to this radial hypha, and then within these cross connections requires the two hypha to decide to fuse together and make back the same organism. But they actually have to go through this self-non-recognition mechanism in order to say, yes, you are me. In case like another spore falls, that is not. So this is, you can't really see this, unfortunately, but this is like a very classic diagram from 2003 of heterokaryon compatibility. And so basically how, siblings of fungi decide to fuse and, or not fuse, if they're the same individual or not. This is not mating, this is just the hyphal fusion. And so if two individuals meet and they are the same and they have the similar proteins and the ligand, there's a couple of, some intricacies here, but if they decide to form a heterokaryon and all is good, then they will, and the two nuclei that are slightly different will flow throughout the mycelial network. But if they are non-similar heterokaryons, they will sequester that cell where they fuse. Like if it's the same species, we're talking about within the same species, two different individuals. If they're not the same individual, they realize after they fuse, and then this non-self recognition, is dictated by these classic immune genes, decides to kill this cell and not create a heterokaryon. And so in 2007, there are some classical het genes that were being discovered in the glass lab and a couple other labs. And a lot of them had this het domain in the protein. I'm showing here on the lower right. You all agree. And, but then one was found to be this tripartite protein that was very similar to an NLR, like the immune gene in us. And so this was found in Neurospora and Serina, which is a model Sorderierales that is, able to go through its entire process, cellular cycle, its biological cycle in lab. So you can fruit it on a plate. It makes a tiny little flask on the plate. You can gather its spores. So you can do like these genetic assays on it very easily. And so it was used as a model organism mostly for senescence for a long time. But then it gained more research on it when this era of mycology was happening, when people were discovering self-non-self recognition and how the mycelium is actually fusing and forming. And so that discovery in 2007 led to this 2009 paper, which is the beginning of thinking fungi as having an immune system, because of this one protein that was found that was very similar to, again, these mammal immune receptors and plant immune receptors. So this is a first figure that shows, how the fungal immune receptor lines up with the commonly known mammal and plants. Yeah, and again, this architecture of these proteins is related to the pathogen recognition receptors, ensuring innate immunity in plants and animals. This is still one of the best reviews out there on the subject, I'd say. Like, Matthew Paoletti is like a very good writer, and there are so many reviews now on this subject, but we still don't have quite a great answer. And I think, yeah, if you want to read one review, you should read this one. And it really also explains this heterokaryon incompatibility, allorecognition, mycelial building in the cellular sense really well. And so, yeah, great, that one NLR in Neurospora was found, and it's like similar to an immune receptor, but like how abundant are these proteins in fungi? Actually quite abundant. And this is the first paper from 2016, so this field seems quite slow rolling, and it's because who cares? Who cares if fungi have an immune system, you know? And so, I mean, I've been at the forefront of trying to get funding for this, and I got one NIH grant when it was a Facts of Life grant, where it was just like funding basic biology, but people care about immune systems if it's plants or if it's humans. You know, save our agriculture, save our humans. Like, we don't really care about fungi, like, whatever, you know. So 2016, finally, a bioinformatics paper was published, like looking through most of the tree of fungi. And very interesting, one of the most interesting findings is that no NLRs are in any yeasts. And this seems to be lost, not like it evolved in multiple clades. And so this is a great paper to read as well. It talks about all the different domains of the proteins. If you wanted to dig into it more. But yeah, this is like half of a figure of the whole entire figure. There's also not very many NLRs in basal fungi. So it seems as if Dicaria has the most. And people hypothesize this because they have the greatest amount of, you know, structured tissue or whatever. You know, there's all these hypotheses, but I mean, hard to say exactly why NLRs are there. NLRs are found in bacteria. There's also hypotheses that they were delivered, you know, through horizontal gene transfer. But I mean, these proteins are very ancient anyway. So yeah, there's a whole diversity of them. For the actual mechanisms of NLRs in fungi, there are only 7 known, and this is all from heterokaryon incompatibility. And so these are the 3 model Sorderieales that Rudy gave introduction on flask fungi. They're all flask fungi, Podospora, Cryphonectria, and Neurospora. And they have all been studied of how they all have been used to study how the mycelial network forms. And these are the proteins that were discovered within that system that are NLRs. Yeah, this is another review that I have at the bottom, but this is a table 5 from my chapter 2 in my thesis, and these are all the reviews and bioinformatics studies on fungal innate immune systems that reviews the components and proposed mechanisms. As you can see, there are a lot, which is great. But yeah, what did I really want to say here? Mostly that, I mean, a lot of people are hypothesizing what these proteins are actually doing, but why don't we have an answer? Like, do fungi have an immune system? These reviews, you know, just sort of give hypotheses. And so going back to the initial question I posed, how are immune systems typically discovered? So it seems like we got homology of known immune components. Definitely have that. We have years of study on that now, like 15 years, right? We know that these receptors are in fungi. We have other immune components like gasdermin known in fungi. And yeah, this is another one specifically on the Sorderieales, because now people are focusing on the Sorderieales because of the heterokaryon incompatibility stuff. But what disease are we trying to cure? Does anyone have known fungal diseases? Can you think of a known fungal disease?
Disease caused by fungi or a disease
Oh, sorry.
for fungi?
disease of a fungus, like something is making the fungus disease.
Candidiosis, aspergillosis.
That's for humans though?
Yeah.
Yeah, aspergillosis in humans, but what is a disease of a fungus? Like what would be a disease of aspergillus?
Yeah.
Maybe the bacteria,
Yeah.
but it's like, yeah, that's a good, yeah, that is definitely the most common one. So hypomyces infects fungi, like it's parasitic on fungi. And a lot of people actually mention that hypomyces is a good example of fungi actually having disease. Like trichoderma. Like trichoderma is a parasite, a very general fungal parasite found eating fungus. But what are the symptoms? You know, like what are we? Yeah, anyway, this is a problem. And like when you talk to microbiologists, they're like, oh, fungi don't have disease, they're just competing with microbes. You know, like because of the way they've been studied, it's just like, oh, they're just in a petri dish, like competing, right? And so the current paradigm of fungal defense strategies comes from academic research based on the discovery of antibiotic and toxic compounds produced by fungus. So this is our most concrete example, is that fungi produce antibiotics, right? And so ever since the discovery of penicillin, which was, penicillium growing with staphylococcal bacteria, they discovered, oh, yeah, fungi produce defense, compounds that we can harness. And so that has really been the starting of how we discovered antibiotics. And that is what is mostly cited when you look, how do fungi defend themselves? They go through the whole list of antibiotics they produce, right? This paper, and a lot of the work done in the Künstler lab in Switzerland, also discovers a lot of antibiotics, because that's how you get funding. You discover a new antibiotic, yay, you get funding. But they noticed that the antibiotic was increased, like 3 or 4-fold, when actually in the presence of the bacteria. And sometimes these cryptic antibiotic clusters that do not express in the lab are only expressed when in the presence of the bacteria. So what is dictating those responses? So yeah, I am like constantly going back and forth with reviewers on this, because people are like, oh, it upregulated antibiotics and that's the defense. I'm like, yeah, but we're looking at how it is upregulated. Like, what is upregulating that? Like, what is dictating the cryptic antibiotic clusters that fungi have is probably the presence of the bacteria they need to defend against, right? Anyway, so yeah, a lot of discovery from Copernopsis of antibiotics, like specifically from actually putting the bacteria on Copernopsis, doing RNA sequencing of Copernopsis, discovering these cryptic antibiotic clusters actually being turned on. Yeah, and actually penicillium increases like 3 or 4-fold when in the presence of the bacteria, even though it's typically on at a low amount as well. There's a lot of fungi that will produce antibiotics like passively, but a lot of these clusters are only turned on in special occasions because they are difficult. It is difficult to make antibiotics. So, yeah. And disease terminology for fungi, like mainly where you see, oh, this fungus is disease, is in fungal agriculture. So all the Agaricus bisporus literature, a lot of it is how do we save Agaricus bisporus from all this bacterial splotch that's occurring. And they don't have great genetics on Agaricus, so they can't discover like how is it becoming susceptible. But they have been able to introduce organisms that can compete against the other bacteria. And so this is also showing the range of fungi do have disease. They have bacterial rot, they have associations with bacteria, but they also have associations with mutualistic bacteria. So this is showing that Pseudomonas crudita is actually a very common Pseudomonas that is included in Agaricus bisporus farming because it prevents disease of other Pseudomonads. And it attaches to the hypha and basically helps the mushroom be healthy. And so very similarly to us and our microbiome, like we have good bacteria, we have bad bacteria, and our immune system is what really selects for what, you know, we want and what we don't. Also how the bacteria behaves, of course, like if it's, yeah, being a pathogen versus not. Yeah, so these two fields, yeah, just, this is what you can do in a lab, is you can have a petri dish and you can add bacteria, and if the bacteria helps the fungus make tissue, it's generally thought of as a mutualism, and if it, you know, decreases the fungus tissue, people think of it as competition, right? They don't think of it as a disease. They think, oh, this bacteria is, so yeah, it's just, yeah. A bit of terminology that we need to really straighten out. So one thing I try to prove through a lot of my pictures, these are all my pictures, is that fungi are host to very specific microbial pressures, and this has existed in the literature for so long, like, and this is something I have to continuously prove to like microbiologists and immunologists that have never thought about fungi, right? They think of fungi in the lab, they think of yeast, you know, they think of competition. But fungi are host to very specific microbial pressures that actually, you know, consume fungal biomass purposefully and not indirectly or passively, or like the sugars that the fungus is releasing. You know, it's not just this competition, it's that they're actually infecting the tissue. And so a very common one, which I have all down here in the left box, is hypomyces and hypocreales. Most of those fungi are actually pathogens of other fungi, and they specifically, you know, infect fungal tissue at certain stages of the fungus lifestyle, and are typically biotrophic to a certain extent. Like they grow with the mushroom and they produce spores on the mushroom, and then, you know, that limits the fitness of the host, right? So it's considered a pathogen because, as you can see, the perifacial Hypomyces lactiflorum that is on the Russell of brevipedes in eye is taking over all of the gills. So Russella is not producing spores, it's the Hypomyces, but Hypomyces, you know, specifically infected the cells. And there's a lot of great microscopy on this now from a lab I've been talking to, but still like really relatively not understood, like when Hypomyces is infecting and how it's manipulating Russell brevipedes. But yeah, also lots of herbivores, specific infections and tumors being formed, tremelaceae, all of them are mycoparasites. There's a lot of mycoparasites infecting other fungi. And there's a lot of bacterial rot. Like, how many of y'all have gone out, grabbed a mushroom, and it's been slimy? Yeah, all of us. Yeah, that's likely bacteria. And that bacteria likely had to interact with it in a small way before it started degrading it, right? So, yeah. Okay, so, yeah, proving through also the literature that bacteria have intimate interactions with fungi, colonizing and manipulating cells. So, when you think of mycelium and a fungal cell, you know, it's this elongate cell with septa, and there are many described ways that bacteria interact. They go inside of the cell. There are many endohypobacteria have been discovered, and they also can attach and exist on the external tissue of fungi. And these are the most common historical examples. This is a specific bacteria, I forget its name, it's just referred to as MRE, inside of Gigaspora, which is a AN mycorrhizal fungus, and it's spores. So without this bacteria, Gigaspora does not produce spores. So now this Gigaspora has this obligate bacteria, and this bacteria cannot live without it. Another common example is Ralstonia, which is a fungal pathogen, can host many different bacteria and chlamydospores. So chlamydospores have now been kind of discovered to host bacteria, be the main source of, not the main, but kind of specialized to host bacteria as we're discovering more and more what chlamydospores are doing. And typically, yeah, this Ralstonia actually helps the pathogenesis of the Fusarium, sorry. Ralstonia is a bacteria, which are little green dots. And the fungus structure, the big chlamydospore, is Fusarium, which is a pathogen. And there's many examples of how they can work and help each other. And there's a lot of examples of how it can hurt. Another really well-studied now system is the cheese-rind microbiome, which is known to be a consortium of fungi and bacteria. And there are some great studies out there. But this one from 2008 shows you, here's the film, and, looking at it, you have these cultures of bacteria, you have bacteria swimming along the hypha. You can see these little bacteria cells all over the hypha, and this is classic for extracellular bacteria colonization of the hypha. They actually attach themselves to the hypha. And there was an RBT and seq, which is a bit complicated to explain, but basically they made knockouts in the bacteria and the whole bacterial genome, and then they threw it at the fungus, and the bacteria that did not survive had proteins that were knocked out in iron biosynthesis and biotin biosynthesis mostly. And so there's very similar to like pathogens of us, there's this limiting of nutrients, there's iron, biotin, but this is like well into the very late stages of this pathogenesis. Very similar to bacteria, fungi have intimate interactions with other fungi. So this is the Hypercrales and Tremelaceae, two very commonly well-known microparasites. Trichoderma actually injects itself into the hypha, typically it wraps itself around, it coils the hypha of its host fungus, and it kills it and eats it. And through like a lot of noxious ways. And Tremella, however, is more of the silent parasite where it makes a haustoria in its host fungus. Here it's Sterium, and it just can live off of the host fungus's nutrients. But it's really unclear, and it's not well known if this actually hurts the Sterium. Is this just more of a passive parasite, or is this is it fine without it? So a big thing that I propose in my work is that we need more model systems to study the fungal innate immune systems. Like we have all these examples from nature. We have like a huge historical body of literature from nature of pathogens of fungi, and then we have this really well represented body of literature on the immune components, like how do we start studying this mechanistically, like genetically. And so these are like a lot of model organisms that already have pre-existing studies of, these are the pathogens and then these are the fungi hosts of like bacteria and viruses and herbivores that are eating them. And there's a lot of ecological studies showing this as well for, many other fungi, but those fungi don't have great genetic tools. It's hard to actually study their biochemistry. And so this is sort of the list I landed on. And I'm gonna go through this really fast. But here at the bottom of this crazy mess is how many NLRs this organism is expected, this fungus specifically is expected to have. Candida albicans is a yeast. It doesn't have any NLRs, but it actually has the most well-known mechanistic interactions with bacteria. And there are many bacteria. This is Salmonella enterica, Serratia marcescens. I don't really know the medical bacteria anymore, but S. aureus and but the most well-known mechanism of how they interact is Pseudomonas aeruginosa. And this is because Pseudomonas aeruginosa and Candida albicans constantly form biofilms in the human lung, and it's actually quite deadly for cystic fibrosis patients. And so studying this mechanism actually is very medically relevant, so it has a lot of funding. So, but only very recently have we sort of discovered all of this. This is, So we knew that Pseudomonas aeruginosa creates biofilms, right? And it requires that the candida makes the hypha before it creates biofilm. So there's all this signaling that occurs before the candida makes a hypha. And this occurs through the recognition of peptidoglycan, which again is this microbial ligand. And it gets imported into the cytosol through these transporters, and then this little tiny adenylate cyclase recognizes it, and it actually has a domain, has half a domain of an NLR on it. And so, yeah. So anyway, this basically, cascades. We know some of the transcription factors now, cascades into hyphal development and the creation of adhesion, so it can start making the biofilm with the Pseudomonas. Yeah, and typically this is actually beneficial within the presence of humans, or within the presence of it, not beneficial for humans, but beneficial for Candida and Pseudomonas to make biofilm together, because then they're more resistant to antibiotics, which increases the likelihood of cystic fibrosis patient to have complications. Yeah, and so I go over this, and this is the beginning of a huge part of my thesis, where I try to accumulate all of the signaling components of these, and this one has a lot of new research coming out all the time, so it's great. But yeah, I recently talked to some of the researchers that do Pseudomonas aeruginosa interactions with Candida, and they weren't even thinking about, that this was evidence of the fungal immune system, and now they're like, oh, yeah. But yeah, no NLR, so if we wanted to study NLR recognition, not gonna happen. Rhizopus microsporus is in the mucormycota. It also doesn't have any NLRs, but it has a really well-known bacterial interaction where this myco-inhabitants can ingress into the hypha, and it makes this mucor much more, mucormycota, much more pathogenic. And so that's how it was discovered as a plant pathogen, Rhizopus microsporus. And they discovered that this bacteria actually is causing a lot of the plant symptoms as it's in the hypha. And so there's a lot of mechanisms of how this bacteria survives in the hypha. It has to constantly interact with something, but we don't have really great genetics in Rhizopus, so we don't have much mechanism understanding from the fungus side. So here in red, which I didn't explain very well, it's all the genes that the bacteria uses in this interaction. Here in black is all the genes that the Candida is using in the interaction. But we don't have very many from Rhizopus. We don't have very many for most fungi. Cryphonectria parasitica was a very well-studied plant pathogen that basically wiped out the entirety of the American chestnut, and it was researched for viral interactions for biocontrol. So they found a virus in Cryphonectria parasitica, and this is the main fungus we understand viral interactions in. Actually, the only one. Now we have more tractability in Neurospora, but yeah, this was mostly researched because the virus was able to slow down the disease in chestnuts. It didn't do a great job, and all the chestnuts are dead. But yeah, we still know a lot of how this is happening. So again, I'm showing the incompatibility. If you can kind of remember, I mentioned that this is a fungus, a Cryphonectria, that where NLRs are in, and we don't actually have a great genome for it for some reason, and so I wasn't able to see how many NLRs are in it. I still need to try to figure that out, but it has NLRs because they've been discovered in heterokaryotic incompatibility, and so right here I'm showing the fusion of two different individuals, and it was proven that this fusion and incompatible reaction actually stops the virus from infecting Cryphonectria, but that's the incompatibility system recognizing itself and not the virus. So it's not really, nothing has been proven, no receptors have been proven in fungi to actually sense virus besides RNAi And so RNAi is another very, very ancient viral defense strategy. And we can talk about it if you have any more questions. Copernopsis has been studied mostly for the compounds it produces. But all these compounds are induced by the presence of these microbes. So there has been 2 nematodes they put on Copernopsis, and they discovered many different lectins that they have, commercialized for anti-nematode stuff, and a lot of proteases against bacteria and lysozymes against bacteria, a lot of different antibiotics, lacnopodin B and copsin are famous examples. And there's some like RNA sequencing that suggests, oh, this cyclic AMP, billet cyclase produces cyclic AMP, oh, and there's these MAP kinases that might be interacting with this, but yeah, like not really well genetics. I mean, not a lot done in Copernopsis to say how these molecules are being upregulated. It's mostly just describing the molecule, which is its own paper, right? And so basically I'm showing the gaps and what researchers need to kind of focus on. Trichoderma actually senses chitin. It senses different chitin of different fungi. And so this is where it gets kind of complicated, because how does a fungus sense a different fungus? Well, there is totally different ornamentations on chitin and different orientations. So apparently, this hasn't really been studied a lot, but these chitin receptors are thought to, they definitely, basically, the fungus that has been studied mostly for its chitin response is Trichoderma, which is the predator. But now people have started to look into the prey, like of the plant pathogens like Neurospora and stuff that, and they also recognize each other's chitin, and there's like very small variations. But yeah, I'm showing the dashed lines because it's not really well understood how these are all connected. But yeah, they'll knock out a gene and say, oh, it doesn't produce this chitin ACE anymore, or it doesn't produce a secondary metabolite anymore, so it must be related, right? But like really not well understood biochemistry of how Trichoderma is recognizing its prey and translating that. So yeah, my main conclusion in this whole thing is that we need to study as many systems across fungal kingdom to elucidate the common fungal immune system. And so putting more microbes on model systems and testing for common immune morphology and phenotypes, which has already been really well documented in these cases. So a lot of these systems just really need to do genetics on to prove that these certain genes are involved. I'll go really fast because I'm like pretty over time on my section 3. So basically I decided to do this in Neurospora crassa. And so I decided I was going to look into bacterial Neurospora interactions. There's a whole library of gene knockouts you can order in Neurospora. So I say, oh, I want to look into this gene and how it functions in the presence of bacteria. I just order it. I don't have to make the knockout. So these sort of tools that these organisms have can really accelerate these discoveries. So I was really young when I started all this stuff, so I didn't get to the spot I wanted to, but yeah, this is showing Neurospora growing alone. This is showing Pseudomonas growing alone. This is showing Neurospora and Pseudomonas growing together. And so you can see from this picture, Pseudomonas is sort of all over Neurospora, but I have a GFP tag on Pseudomonas to prove it. Even though like clearly this Neurospora is doing slightly worse than this Neurospora, but I'll still go through the reviews and be like, oh, it's just competing for nutrients, and I'm like, it's infecting the cell. But yeah, this is E. coli, and this is E. coli on Neurospora. And E. coli can swim in this environment, but it doesn't swim along the hypha. It is causing a different Canidia phenotype for sure, but it stays locally in the center. And so when I first started grad school and I first decided, okay, I'm going to study Neurospora, I asked, does Neurospora have any bacterial interactions already discovered? And there was this one paper, which is actually cited a lot in fungal immunology literature and research, that there is this one gene from Pseudomonas that can induce programmed cell death by, interacting with this heterokaryotic compatibility system. And they showed that Pseudomonas was able to traverse along the hypha extracellularly, and it replicates as a sole nutrient source. So they grew out Neurospora on plastic, and they put the bacteria on the hypha, and they saw the bacteria swim along the hypha with no other nutrient source besides, plastic, which doesn't eat plastic. And so this is crazy because this bacteria is from a tomato plant. Like, this is a pathogen of a tomato plant. Well, yeah, Pseudomonads are typically like very general pathogens, and this Pseudomonad has been shown to infect many different plants. So, it wasn't too far off that it was infecting a fungus that grows on plants. So Neurospora's natural habitat is burn habitat, and it canidiates after a burn all over plants. And so, most likely the common bacteria that are around Neurospora are also around plants. The main point of this paper was they ectopically expressed this gene. This is without bacteria, but when they put the gene in Neurospora and they induced its transcription, Neurospora clearly does not do well. This is Neurospora without the induction of the gene, and this is the killing of Neurospora. However, the bacteria that didn't have this gene, kind of caught up with Neurospora, like with its wild type component, and so this gene is debated if it's actually doing anything in this. They basically had to force this gene in Neurospora to prove that it could cause cell death, but it doesn't actually seem to get into the cytosol during the interaction of the bacteria growing on the fungus. So basically, there was a lot of gaps in this research, and I was attempting to fill it. So my first question was, does Pseudomonas actually interact with Neurospora in native environments? Like, is this actually a system like I want to develop? Both the bacteria and Neurospora had genetic tools associated with them, so it would be easy, but like they're not ecologically relevant. But this is how we discover immune systems all the time, because we're using mice and we're using human pathogens. And the mice is not, natively like interacting with humans. But you can kind of force these interactions to try to figure out the molecular biology in a way. But I wanted to make it ecologically relevant, so I basically went to France, and where the Neurospora European clade is found that is closely related to crassa, and I sampled a bunch of crassa from burned habitats, and I took as little Canidia as I could, and I put it in the center of a plate without antibiotics, and I saw what bacteria grew out with Neurospora. I also did sequencing of the wood chip itself, but that is data I have not analyzed because it was too much. But maybe one day in a paper. So I sampled 87 distinct wood chips of Neurospora, and out of the 87 I sampled, 25% grew out with extrahypobacteria. And so 5 of these 25% were Pseudomonads, and a lot of them were plant pathogens, like Erwinia and Pantovia. And so this is just an example. Most of them were only infected by one bacterial morph. Like I would swab the whole hypha onto a plate, and I would only get 1 or 2, mostly 1. And so a lot of them were Pantovia, and they like grow out like, kind of what I saw you see in the cheese rind microbiome paper, this like glossy mycelium. And the mycelium is still growing. It's growing just with bacteria. And this is Stensomonas spencerii. It made these like more like harder biofilm pockets on the hypha, which is interesting. And these are all bacteria I hope to really, really study. But I basically proved the ecological relevance of the system, and I decided to dive more into the system. So this is showing, again, the GFP fluorescence of this first picture. This is showing at high bacterial inoculation versus very low bacteria inoculation at very similar time points of Neurospora, and how the bacteria swims at different bacterial inoculation. Basically, it hurts conidiation, like clearly you can see, I use an image processor to prove this quantitatively, but can't really see it, but yeah, Pseudomonas basically, when it's infecting Neurospora, when it's all over the Neurospora hypha, the conidiation does not occur, and then that's in purple here, down here, and this is in two different environments. So this is Loria agar, sorry, I could have used the cursor the whole time. And this is Vogel's, which two different environments. Vogel's is what Neurospora enjoys the most, and even in this environment, the conidiation, as you can clearly see, is not as pink. And it's interesting because I didn't see like GFP fluorescence of any bacteria, but clearly, yeah, I mean, I had to see a lot of bacteria to see the fluorescence, so it could still be there. Just kind of hard to see when the mycelium is super robust. I basically proved, which was not shown in the first paper, like any fitness on the fungi. There's no fitness measurements of fungus. Like that's a problem. Like how are we going to describe that this is a disease versus not if there's no fitness? So here I have inoculated E. coli, which should never have seen Neurospora. E. coli is in our gut, so it's not considered like a possible pathogen, right? It's still, when inoculated onto Neurospora at the highest amount, this is highest to lowest amount of bacteria. This represents like 1 or 2 bacteria. This represents like 100,000. At the lowest amount, at the highest amount, sorry, E. coli is still doing pretty, it's not doing as well as Neurospora alone, which is in the blue. Pseudomonas, however, really decreases the growth of Neurospora at the highest, and even at the lowest inoculation had a significant difference in the fitness, and that's like 1 or 2 bacteria on Neurospora. And so this is just, robust quantification of actual whole organismal fitness, which seems to be really lacking in these bacterial fungal, papers, because all they care about is it produced this antibiotic. Yay, that's the paper. So anyway, microscopy of the colonizing phenotype shows immediately the bacteria surrounds Canidia and germlings, and it typically grows out with Neurospora for some time, and then Neurospora is very fast, so the hypotips will grow without bacteria for a while, but where the bacteria is 24 hours prior, there's then typically a robust biofilm. And so, yeah, this is just showing the colonizing phenotype. This paper also is online, so maybe I should just rapidly go. But this is kind of a crap confocal video, and I always meant to redo this, but you can still distinguish that some of the bacteria are attached to the hypha, where there's a lot of free flowing bacteria. Eventually they will all become attached to the hypha if they can, or be in a biofilm. This is before the biofilm. There's some bacteria stuck, and there's some swimming around. And they're typically stuck, not polarly, but with the whole bacteria body. Anyway, so a major finding from the paper is that the spread, this is showing that the spread of the bacteria is inversely correlated with the development, and actually how fast Neurospora grows, which is, I skipped this, which is if Neurospora is allowed to grow and it doesn't have a strong bacteria inoculum at the beginning, the Pseudomonas grows faster. And so an observation from the previous paper is that this bacteria is not killing the cell, it's actually keeping the cell alive. And so it seems as if the bacteria wants Neurospora to be alive. And so if Neurospora is doing better, typically the bacteria is spreading further. And so this is just kind of proving that the bacteria, I mean, Neurospora is clearly still doing not the best. It's diseased, but there is some sort of manipulation that the fungus doesn't just die immediately, right? Which is debating if there's like a toxin or whatever. The main finding is here for this graph. This is hypha growing without bacteria. Or sorry, this is hypha growing with bacteria, and this is like the wild type bacteria, and this is hypha growing with a bacteria that has a secretion system knocked out. And so this is showing that the secretion system might be important for the colonization of bacteria. This is a lot to explain, so yeah, come read the paper if you have more questions, but I also proved that cell death is not happening, corroborates the finding of the previous paper, even though the whole paper is based on the fact that this one protein can cause cell death. I was just looking at the germline stage, and I was doing flow cytometry for a long time to prove this, but you can also just prove this by doing CFUs, and so clearly not dying, but you know, I have done a lot of microscopy and I haven't quantified it, but there are a lot of dead cells and bacteria that seem dead around it. So the red is a dye that only goes into cells and fluoresces if it can. And so this is when the cell basically has holes in its cell wall, and that's usually indicating the cell is dead. And so this is showing, yeah, there are segments of the hypha that seem to die, and then around them there are dead bacteria, which is interesting, but still doesn't prove anything, and it's hard to quantify and like do genetics for, because I'd have to count microscopy cells forever. So I tried to solve this with flow cytometry, and it's a method that I created that's part of my thesis that can help if there's an actual bacteria that does cause cell death, but this bacteria is not causing cell death. I did RNA sequencing on Neurospora as soon as I put bacteria on it, like 10 minutes and an hour after bacterial exposure, because immune systems are very rapid. And so if there's like sort of immune genes and immune things happening, it would be in the very beginning. I saw that like 17% of the genome was significantly differentially expressed once I put a lot of bacteria onto Neurospora. And I clustered all these genes based upon expression level, and I decided upon one cluster that was interesting to me, which had this lysozyme that is a known antibiotic that chews up bacterial cell walls. And within this cluster, there's also a couple other copper transporters and ferric reductases, superoxide dimutases, these things that cause these reactive oxygen reactions, and this multiple drug transporter. You know, all of these things that could be detoxifying the cell, but they're rapidly turned on in the presence of bacteria, or putting antibiotics out there. I promise I'm almost done. These are the genes. This is wild type Neurospora at 72 hours when the pink is infected with bacteria, and these are the blue is just Neurospora without bacteria, and each of them are different because they have had a gene knocked out. So if Neurospora is doing well even without these genes alone, but in the presence of bacteria without these genes, Neurospora is doing significantly less well than the wild type. So this sort of infers that these genes might be involved. And I also had an NLR that was downregulated. It seems to be doing a little better, but it's not significant. So yeah, this interaction probably is not involving NLRs, and I am really curious to continue the work on the other bacteria that might be involving the NLRs, and actually, you know, looking for intracellular bacteria of Neurospora, and that's kind of where I'm leading into my postdoc research. And so this is an overview figure of my RNA sequencing and some of my genetic testing I did for this paper, and possibly like what is immune signaling, what is not. Typically people, when they discover immune systems, they find one gene output, and then they can trace it back to what's recognizing it. So there's all these antibiotics, the secondary metabolism that are turned on, and all these things I could continue to chase to try to find receptors, but that's for a different paper. Because I had to actually prove all the fitness stuff first. So thank you for listening to me ramble for a long time. Yeah, I'm working on Latoporus here in LA with the company. She actually came to a couple of y'all's events, the CEO of this company. Basically, we're trying to fruit Latoporus. Well, we are fruiting Latoporus, but my job is to make it fruit better. So I'm doing a lot of like nutrient testing and stuff, and it's been really fun, like classic mycology, like figuring out how this fungus likes to grow, like in culture, and actually fruit in culture, which is great. And so yeah, I'm glad to be in LA, and I'm looking for more community always. So hit me up. And yeah, acknowledgements. This is my grad school advisor, Christina. This is Louise Glass, and they both put in me because Louise really helped me during my PhD, even though she was retired. So they both came for me, and that was awesome during my graduation. Monica is a professor in Vancouver now, and she just studies fire fungal ecology, and yeah, she's the one I burned down the forest with and wrote that paper with, and yeah, she's a constant inspiration. And then this is Jude. She is taking over my project, so hopefully, even if I don't continue with this project, there'll be more info coming out. Ethan and Nathan helped me create the mycology club at UC Berkeley, which actually became quite robust and was super fun. And what happened? Yeah, I just had a great time when I was in Northern California. So this is my acknowledgement slide. And Warren taught me how to identify a lot of mushrooms. So I thank him too. Okay. Anyway, yeah. Thanks.
We have time for questions if you'd like to take some.
I'll take questions.
I have a question about the homology and the NLR receptors.
Yeah.
How deep does that homology go in the cases that it goes deep?
Yeah, so the N-terminal part of the protein and the C-terminal part of the protein are quite diverse. They have the N-terminal is like the signaling domain, and the C-terminus, that domain is involved in recognizing different pathogens, and so that is what evolves. It seems like this protein, that evolves very rapidly, whereas the oligomerizing domain, which is a center domain, is very ancient. It's like found in bacteria. It's quite conserved. It's the stand ATPase. It recognizes, basically it's this sensor that typically has a repressor on it, which is the C-terminus. It's like folded like this. And so when the ligand comes, it opens, and then that signaling domain attaches to like 4 or 8 signaling domains of the same protein. And it is an HPase, so it gets a phosphate from ATP to start its reaction. That's the conserved portion mostly. Yeah, and that's what people use for bioinformatics studies. The repeat domains are very similar. Like they have very similar coiled structures. Like there's this, alpha fold. The folding of the protein seems to be conserved, but the sequence is not conserved. So now with like AlphaFold and other AI ways of folding proteins, we've figured out like geometric homology as well as like sequence homology, and it seems like those two things are conserved even if the sequence is quite different. The sequence also dictates what it binds to, so that's why it's different. But that is found in bacteria, that stand ATPase. There's 2 versions of that have evolved, and it seems as if For plants, there's, yeah, I can show you. But yeah, so this middle domain here, this knocked in the art, knocked, these are the two main types of this middle domain. And the knocked is found in bacteria. The indie arc seems to have evolved at a later time. I mean, it's really complicated because these proteins are actually some of the most well-known horizontally transferred proteins as well, because they are receptors for like recognizing threats. So it seems as if, yeah, it's sometimes hard to say, but yeah, no, for the longest time, there's actually a paper here. It is comparative genomic analysis reveals an absence of leucine-rich repeat pattern recognition receptors, proteins, and fungi. So this paper in 2010 actually led to us believing there was no NLRs in fungi, because at that time we were just looking for the LRR domain, which is this, yeah, before we realized that the most conserved region is clearly the stand ATPase in the middle. And that, I mean, in mammals there are other C-termini domains, like WD40 is what is shown here, but yeah, all these other folds, but there are these repeat domains that tend to coil in a certain way. And so they all have very similar like architecture after they're made into proteins, but they're quite different in sequence. But yeah, this is quite conserved. Homology, that's how people find them now doing bioinformatics, is they make an HMM of these regions, the NBRC and knocked. Yeah. Yes?
So I had a question, and correct me if I'm not understanding correctly, but it seemed like in the last section where you talked about your research, you mentioned just that the presence or absence of NLR didn't seem to have that much effect, at least on the studies that you did on the success of the fungi.
Yeah.
How comprehensive was that? Was the, I guess, the identification of NLRs in those studies, and is it possible that there were other NLR genes that were there that were affecting some of the others?
Yeah, the only NLRs I looked at were in my RNA-seq data set. There are two downregulated in the presence of the bacteria. So, I mean, NLRs are highly related to cell death in many cases. Like, a lot of them can just signal, but a lot of them typically signal apoptosis. And so I hypothesize that NLRs would be more important for an organism going intracellularly. Not to say that Pseudomonas isn't interacting intracellularly, because clearly the type 3 secretion system is like secreting things into the cell. And that's probably how it, there's proof from my paper that helps it colonize, right? But there are 17 NLRs in Neurospora. And actually the main thing now that the grad student is doing after me is like looking at all of them with many different bacteria pathogens. Because, yeah, I mean, I wanted to holistically describe like the reaction it's having and not just focus, because that's a bias, right? Focusing on the NLRs, like, because I wanted it to be as unbiased science as possible, but people want that. People want to see what the NLRs are doing in fungi, and so people don't care as much. They're like, oh, yeah, but you have to first find the bacteria that's actually interacting with an NLR. So, I mean, in this case, it seems as if Pseudomonas just takes over Neurospora. So, and this is just one Neurospora. Like, I need to, my postdoc proposals are looking at the whole population of Neurospora clade B, because there might be some that are resistant. And the NLRs are all different across each individual Neurospora. So it could be that this Neurospora is just susceptible. And so maybe the bacteria is downregulating the NLR. Yeah, who knows? But I mean, the fact that the NLR showed up in the RNA-seq data set means that there is some sort of process of immune systems happening. There's also all of these proteins I discovered, which I didn't put in this paper and I really should have, but they're called NAIP proteins, and they're specialized at knocking down NLRs. Because yeah, NLRs really only should be used if they're really needed. I don't know. The answer to your question is yes, it could be a different NLR, but it's also probably a different bacteria or a different Neurospora species, individual. So yeah, the other way to do this is to find all the bacteria, a ton of bacteria with Neurospora or viruses, and then a ton of different Neurospora, and score them resistant or susceptible, and then mate a susceptible Neurospora with a resistant Neurospora, and then check the progeny, and then you can do mapping back to the genes that are causing this susceptibility and the genes that are resistant, and that will give you an unbiased approach. And that's what I proposed to do, but I didn't have the resources, and now I have the resources, so hopefully someone will give me a postdoc. But yeah.
So just a follow-up, because it was interesting to me that when you were talking about Candida applicants and that that it lacks NLRs?
Yeah.
Or at least, and so in my mind, so like for in a CF patient who is possibly who's more likely to be colonized with Candida and with Pseudomonas, it almost seems like it could be like a protective or like a selective factor that it allows it to allows them to live together without having apoptosis in the presence of Pseudomonas.
Totally. Yeah. I mean, the main hypothesis that people say why yeast lack NLRs is because of this cell death reaction again. Because if the yeast kills itself, what is it protecting? Nothing. Cell death equals organism death. So typically single cell organisms lose NLRs because it's not helpful anymore. Yeah, exactly. So if it just killed itself in the presence of Pseudomonas, I mean, it seems like in culture, Candida does worse in the presence of Pseudomonas, but within humans it does better because it can protect itself with biofilm and stuff. So it's all complicated, but yes.
I think you actually kind of just touched on it. I did want to clarify the biofilm creation that you mentioned as far as Neurospora when it's infected with Pseudomonas. Is that a defense mechanism of the Neurospora, or is that a result of the bacteria?
I think it's a result of the bacteria. I'm sure.
The bacteria creating it?
Yeah, the bacteria creates the biofilm. I'm sure. I mean, based upon Candida and Aspergillus studies, it seems that the fungus also produces adhesions and certain things that go into the biofilm, and they produce it together. But I mean, based upon my microscopy, like all the biofilm is green, which means that it's mostly made of bacteria, but yeah, probably fungal components. It definitely has to dock onto the cell somehow, so that's another thing, and then create like this extracellular matrix of gloopy stuff. Yeah, it's mostly coming from bacteria. And that is like the main, bacteria make biofilms on many things to protect themselves. It's not just fungus, but typically when they make biofilms on a cell, they have to actively interact with that cell. So, yeah.
I just want to say thank you for letting me know trichoderma is not only something we compete with, because I like to grow fungi. Now it's a predator as well.
Yeah.
I'm like, oh man.
Yeah, I mean, I have, like, working on Laetoporus now, trichoderma, and I saw this with Symphycillium as well. I was helping Lotus Lofgren, like, clean her cultures, and she had this hypocreales thing, like, wrapped around, like, it's really a smaller, like, hypha wrapped around, and it will produce, like, 2 or 3 conidia, like your cultures might be infected and then it will like, sporulate, and wherever on the plate, like there's no fungus, there'll also be a trichoderma culture. That typically comes from the fungal culture already being infected, or, from the air because it's everywhere.
Yeah.
And people are spraying it in agricultural fields too, so it's like super abundant. Yeah, it's a biocontrol for plant pathogens, for fungal plant pathogens. So they'll spray it and it helps plants, but it hurts fungus. Yeah.
Wow.
Yeah. It also helps the soil, I think, but yeah.
Are there any components within like the liquid in guttation that protect against pathogens?
Can you say that again? Liquid what?
Like, guttation?
Oh, the
liquid that
guttation,
comes
yeah.
out. Is there anything that's like pathogenically defensive or?
Yeah, I see infected like polypores producing guttation around hypomyces all the time, and I'm sure it's like an there's a lot of antifungals and antibiotics that are in that. And a lot of it is also other metabolites, but in just excreting water, but from their central metabolism. It's like breathing and sweating, but a very common mechanism that's turned on in the presence of predators is excretion. Across all the RNA sequencing, they upregulate all of their transporters, like cellular transporters, and definitely antimicrobials and stuff. So, is that proven? I think so. I think in the guttation there's a lot of antimicrobials and stuff, but yeah.
Outside of Agaricus spores, there are a lot of broader evidence for primordial formation being immune response.
Ooh, you mean like in the presence of the helper bacteria?
Well, you mentioned the bacteria, it's more of a mutualistic symbiosis in that case.
Yeah.
But in the broad sense,
Right.
do mushrooms form because of
Yeah.
these interactions?
Yeah, immune responses and sexual reproduction and stress seem to be very linked. Mutualisms tend to actually interact with the immune system more because they have to prove that they're good and they have to continue to exist there. And so there's, I don't think there's evidence that the primordial formation is an immune response, but I see MAT-A, I see all the pheromones upregulated in the presence of bacteria, and this is also very common. Sexual reproduction is linked to stress, and so like an organism, like a fungus, it's like, oh, it's drying out, I'm gonna fruit now, so I can get my spores out there into the resting stage, and that is typically linked to some stress response, and a lot of these organisms are instigating that stress response, and so people oftentimes see sexual proteins in immune responses too, upregulated. But I don't think it really goes forward with most of, with Pudita, I'm sure it does. But I really need to look into the Agaricus bisporus literature a little more because I do think they have RNA sequencing stuff done, so they have ideas of what's happening with that interaction, but they don't have concrete evidence of an immune system. If that makes sense. But yeah. Any more questions? Okay. It's pretty great.
Let's all give Grace a round of applause. We'll still be around for a while, and we are cleared out of here by 10.